name is Tom. These are my friends. They are going to explain you about the traditional and new biotechnology in alcoholic beverage fermentation.
Among all the fermented foods, the oldest are probably beer, bread, and wine. Although there are differences of opinion about the country of origin, most anthropologists believe that the Sumeric Empire, the Babylonians, and the Egyptians all have a grain-based fermented beverage. This can be traced back to approximately 4000 BC. Alcohol beverages are defined as beverages that contain ethanol. This ethanol is almost always produced by fermentation, the metabolism of carbohydrates by certain species of yeast under anaerobic or low oxygen conditions. Beer, as we know it today, is made of malt, as well as other sources of starch such as corn and rice, water, hop, and of course yeast as the fermentation agent constitute the rest of the basic brewing material. The purpose of the brewing process is the conversion of starch contributed by the malt and the adjunct corn or rice to the maltose and the dextrin.
The making of alcoholic beverages. The beer making. Firstly, malting. The process to allow malt, usually barley, to sprout. It involves 3 steps: steeping, germination, and kilning. Firstly, steeping. The grain is soaked in a vat of water for about 40 hours. Secondly, germination. Grain is spread out on the floor for about days to form rootlets. Thirdly, kilning. Germination is halted by drying the green malt by heating gradually under high temperature. Dried malt is stored in silos. Other ingredients such as corn, rice, and wheat are added to give different flavor, color, and aroma. The second step will be milling. The malt is crushed to break apart the kernels and expose cotyledon. The third step is mashing. It is to turn malt into liquid. Milled malt is then heated in warm water in a mash tun. Starch is dissolved in water, converting it to sugars. The sugar-rich liquid is now called malt. The next step is brewing. Grains are filtered out and wort is ready for boiling in kettle. Hops are added as flavoring agents and the purpose of antimicrobial. The fifth step is cooling. The hop is filtered out from the boiled wort, and the boiled wort is cooled by passing through the heat exchanger and then transferred into fermenter. Cooling the wort before adding yeast is to prevent inviability of yeast cell. Next is fermentation. Adding yeast into fermentation tank. The yeast cell utilize sugar in wort and convert into alcohol and carbon dioxide. Different strains of yeast determine the character of the beer. There are 2 types of fermentation for beer. The top fermentation. Yeast cells such as Saccharomyces cerevisiae rise to the top at the end of fermentation and then skim off, which is usually produced early. As for the bottom fermentation, yeast cells such as Saccharomyces carbogenese sink to the bottom, usually produce lager beer. The 7th step, which is maturation. The beer is racked into new conditioning tank for aging. Taste ripened and liquid is clarified. Lastly, the finishing. Beer is filtered and carbonated. Further filtering finds sparkling clarity. Beer is moved to holding tank for storage or packaged into bottles or cans. Next, we'll be talking about beer production issue and solution. Firstly, low protein haze control and low purity of protease. The solution is Genetic engineering figured out suitable and high-purity microbial protease from Candida olea and Pichia pini. Protease added to prevent chill haze and increase its stability by adding silica hydrogels, polyvinylpyrrolidone, and tannic acid. Secondly, limited enzyme in traditional way. The solution is new biotechnology improves the quality and quantity of enzyme. Amylolytic enzyme is to facilitate starch and dextrin hydrolysis. Beta-glucanases improve filtration and haze reduction. Alpha-acetolactate decarboxylase speeds up maturation. Enzymes such as celobiose, lactose, and xylose allowing to ferment more type of carbohydrate as energy source. Improve alcohol acetyl transferase activity to produce more acetate ester to enhance flavor. Thirdly, wild yeast causes contaminants. Wild yeast will form scum. The scum formed from the multiply of yeast which became heavy branch and rise to the top to form a thick layer. This will trap gas bubble inside to build up pressure and finally force the fermenter to stop to work for a few weeks. This contaminant will also cause the loss of alcohol. The solution is production and secretion of zymosin to control the potential of wild yeast contaminants, or genetic modify the new strain called killer yeast strains to control the growth of wild yeast. Last but not least, other contamination. The solution is production and secretion of nisin from Lactococcus lactis to control contamination by lactic acid bacteria and the de-repressed strain to overcome catabolite repression and speed up the fermentation. As for wine, most of the wine issue can be solved by using recombinant DNA technology. By using recombinant DNA technology, the wine yeast strains will have beneficial characteristics such as production of desirable organoleptic compounds, metabolism of a large portion of malic acid, low urea excretion, improved fermentation efficiency, suitable flocculation characteristic, controllable ester formation, protease production, production of inhibitory substances against spoilage of microorganisms. As for example, nisin to kill gram-negative bacteria and zymosin to kill wild yeast. Pectinase production to improve wine filtration rates. Production of glucanase that release bound terpenes to enhance aroma. And lastly, greater dihydroxyl acetone phosphate reductase and glycerol phosphatase activities to increase glycerol levels.
Okay, there are a few issues that need to be discussed for the new biotechnology method for the production of products. The first one is about labeling issue. Currently, no federal agency requires food to be labeled as genetically modified. One reason is because the agency sees these products as substantially equivalent to traditional food products. This assumption has been supported by courts. And the next issue is religious. Religious groups are concerned that food might contain genes from animals such as swine prohibited by some religions, and they maintain that they have a right to know if food contains those genes. Both Orthodox rabbis and Muslim leaders have ruled that simple gene addition that lead to one of a few new components in a species are acceptable for kosher and halal law. However, cloning in particular raises serious ethical issues for religious leaders of all faiths. Consumers fear that introduction of gene into new supercrops can cause allergies or harmful to human health. For instance, if new genes inadvertently cause a plant to produce toxins at higher level than are present naturally, there could be long-term health consequences for humans. Some consumers are worried that a gene introduced into plants to protect against pests could also cause the plant to alter its pollen, thereby affecting the health of humans prone to some sensitive